last time we were talking about brain stimulation, particularly TMS, Transcranial Magnetic Stimulation, and we started to see how this technique can be used to perturb brain activity and cause, for example, interference of impairment in the behavior. So I share with you a couple of videos in which the were researchers, scholars perturbing brain activity and impairing behavior, which we people has called this approach the virtual lesion approach, because in principle, by interfering with brain activity, in principle, we can demonstrate that a given brain area is critical for a specific behavior, showing that, simulating somehow the effect of a real lesion. Of course, this is, as you will discuss in a bit, this terminology, virtual lesion, is not a real appropriate way of describing, but it gives at least an idea that we can interfere with brain functioning. So last time we discussed a little bit about this. With this brain lesion approach, where my lab and several other labs have tried to to test different brain region in different brain process. This is just an example of sort of review of different studies in which perturbing a specific region that we typically see active during specific process. This is just an example from the action perception literature. For example, the slide, the panel on the left shows, for example, which are the brain regions that are active when we observe another person action. And on the right, you see, now is an old, I would say, an old review merging different studies. Now there are many other studies basically confirming this first result and showing that, for example, by perturbing those brain regions that are active, typically active when we see other people doing an action, when we perturb these regions, we are able to induce reduction in the ability to recognize, identify, discriminate, or discriminate different actions. So in principle, demonstrated that those regions are not only active during action observation, but are critical for that process. Just to provide some example, for example, we also use this approach to interfere with videos like this and asking people to, for example, discriminate whether this is an authentic or a fake smile. And we perturbed different brain regions, for example, the face representation in the premotor cortex or in the somatosensory cortex, because the idea is that when you observe a smile like this, in order to make an inference about the amusement that the person is truly feeling, you have to simulate facial expression in your brain. So you need to activate sensory motor region, and we try to interfere with these sensory motor regions during a task like this, and also a control task. For example, we ask a participant to observe these videos and to see whether the lines appearing in a position close to the eyes or to the mouth was appearing up or down relative to the eyes and to the face. So, a sort of attentional task, spatial attention task, in which participants had to monitor the very same visual features, face features that are important for discriminating real and fake smiles. And and here you can see that interfering with, for example, a ventral frontal side, the inferior frontal gyrus near the ventral premotor cortex, or the ventral somatosensory region, basically in the localization of the face area in the somatosensory cortex, was able to interfere with the smile authenticity task, discrimination task, but not with the other task, demonstrating that these regions are causally relevant for making this discrimination. Well, this so-called virtual lesion approach actually can provide also very useful information about brain organization in different populations. This is a seminal study that was carried out in blind people and sighted people that were able to read braille text. So with haptic exploration of a text, they were able to read, basically, to identify those characters and read entire sentences and so on. So with a group of scholars started from some evidence in neuropsychology, where, for example, there was a blind woman that after a stroke in the visual cortex, she started to be unable to read from braille. So before the stroke, she was able to read. She was a proficient braille reader. And after the stroke in the visual cortex, this woman was unable to read. I mean, so the scholars started to hypothesize that the visual cortex was critical for blind people in order to be proficient and read braille text. So so there were some brain imaging studies, for example, the study by Leonard Cohen, showing that this is a PET study, but this has been replicated also in other conditions. And this study showed, for example, that in blind people and also in the sighted people, when people started to read by haptic exploration of the braille text, they showed activation in the sensory motor cortex, contralateral to the hand that was used for reading, and also a visual activation. But as you know, showing a brain activation doesn't mean that this activation is critical for behavior, and TMS was used in order to interfere with this region. And for example, it was shown that sighted people in particular showed an enhancement in error, show more errors when they were stimulated over the sensory motor cortex. So during haptic exploration, of course, if you interfere with the sensory motor cortex, you interfere with the initial processing of the somatosensory processing, tactile and haptic exploration. So you basically interfere with the initial processing of this information. And sighted people were particularly unable to interfere with this stimulation. Of course, also blind people that are represented in red show some increase in error rates, but sighted people were more interfered. On the other hand, you can see that by stimulating the visual cortex, the visual cortex, blind subjects, blind participants were much more interfere than sighted people, because probably blind people tend to use the visual cortex much more than the sighted people when they have to transform haptic information, tactile information into a more, well, kind of visual format, in another format, let's say. So in order to read, they use that cortex much more than sighted people. So this is a very interesting example of how virtual legion can provide the information about brain organization. But interestingly, brain stimulation interference can even establish not only whether a given region is important for a function, but also when this region is important for a specific function. This was shown also in very early study, for example, this one by Amashan, who demonstrated, for example, that interference with visual cortex during perception of letters or symbols that were flashed for just a few milliseconds. Well, this visual identification task was interfered by the stimulation of visual cortex, but only when the interference was provided around 80, 100 milliseconds. So basically, when the visual cortex is processing, probably is just the visual cortex in that specific moment, so 80, 100 millisecond, is basically is already receiving some feedback information to the visual cortex. And in that particular moment, the visual cortex is making some process which is truly important for behavior, relevant for behavioral, so for behavioral performance, so recognition and etc. So this was quite another important evidence that has been replicated in many other studies testing the chronometry of cognitive process. So temporal information is quite important. And so you can really see how these approaches can be really powerful, because you can test models of cognitive functioning, and testing whether a given brain region is important in a given moment, but not in another. For example, in this particular case, you will see that the visual cortex was stimulated, but after, let's say, 20, 40, or 60 milliseconds, basically performance was not really affected by stimulation. And also stimulated the visual cortex maybe around 140, 160, and so on milliseconds after the presentation of the stimulus. That this interference was not relevant anymore, because all the visual process as was already carried out in the critical time window, which the visual cortex is important for that process. So in principle, I mean, these and many others, many other evidence show that, I mean, you can use TMS to interfere with different brain processes. Here you can see just some example. So attentional, perception, motor function, language, working memory, or executive function in general, space processing, number processing, decision making, and so on. I mean, so the processes are really impressive. So there is really huge literature now showing that you can use TMS in order to interfere with these functions. But not only interfering. In principle, you can manipulate brain activity in order to make these process even even working in a more efficient way. in principle. And indeed, it has been, now there is really growing evidence that suggests that the term virtual legion is not, well, is a, I mean, is a useful heuristic to describe the fact that typically we can observe interference effects. But this metaphor is just a metaphor and does not represent the mechanism of induced effects. So which are the mechanism of TMS induced effects? Well, there are several proposals. One very convincing proposal is the following, that TMS can be conceived as a sort of injection of noise in a stimulated area. So TMS injects noise in a given targeted area. So because this is, I mean, this idea um, underlies the fact that when we stimulate, when we inject noise in a given area, in principle, this noise does not necessarily mean that we are interfering with that activity. Of course, we are creating some physiological effect. In most of the case, this effect competes with neural resources, with the neurons that are solving the task. But in principle, we can conceive this noise as a sort of interacting effect, that something that interacts with the neurons that are solving the task. And in principle, this can slow task executions, but not only hindering task execution. And this is particularly true if you consider, this is particularly relevant if you consider what happens particularly in our perceptual systems. So we know, for example, that during perception, during sensory processing, perception processing, our sensory channels and also our brain are basically embedded into the noise. So there is some noise in all levels of information processing, from receptor, from the activity of receptor to our behavior. So each level is really embedded into noise. But in principle, noise does not only mean that, so having some noise does not mean that we can only impair performance. So in principle, when we, for example, try to process information, the information that some signal that is weak, so at say threshold level, in principle, if we introduce noise in the system, the optimal level of noise, we can even perceive better that stimulus. Please I give you an example here on the right of the slide, where you can see a weak signal. That is, for example, this is the Colosseum. So I came from Rome, so this is just a little souvenir from my city. And this is the Colosseum, and here you will see on the bottom, you will see a very very weak signal, where you do not really recognize what was the picture is showing. On the top, on the other end, you will see a weak signal plus a lot of noise. And also in this condition, perception is not optimal. In the middle panel, on the other end, you will see a weak signal with an optimal level of noise that probably helps perception, helps us to recognize that case with the Colosseum. So in principle, an optimal level of noise that can be introduced by TMS can explain some sort of paradoxical facilitation that has been demonstrated many, many times in the TMS literature. I will provide you an example here. Uh, this is a study in which participants receive a stimulation of the visual cortex, in particular on the V5 region. V5 is a middle temporal region over the posterior part of the brain that is involved in perceiving motion information. So and when we have a lesion in that region, patients are unable to see a moving environment. They tend to perceive like static frames with some break between them. So they have a really chaotic perception of the world because they just tend to see some flashes. And so we think that change a lot in just a few milliseconds. So V5 is an important region for perceiving motions. And in this task, participants were presented with some dots moving in a coherent way or in a highly coherent way, and you can see them here. Hopefully you will see my the mouse of my desk. Here you will see several dots moving all in a specific direction with with just a few dots moving randomly in different direction. And this is the high coherence condition. On the left panel, you will see the low coherence condition, in which all these dots were moving in a kind of random fashion, with just some low percentage of them moving coherently rightward. Okay, so we have a low coherence, near threshold, and a high coherence, suprathreshold motion stimulation. All right. And here you will see, for example, when the coherence was low, participants overall were very, they were not very good at this task. Why, when the coherence was high, they were very good. All right. And in this study, participants received, say, sort of baseline stimulation with no TMS at all in black, and three different levels of stimulation of the V5 target area. So low TMS intensity, medium TMS intensity, and high TMS intensity. Basically, on the left, you will see that, let's look at the right. So in the high coherence, so what does it mean, high coherence? It means that the signal is kind of strong signal. So we have a good signal participant. So in that condition, if we inject noise, probably perception is already very good. And if we inject noise, this can hinder performance. And this is exactly what the researchers found. For example, by stimulating the V5 region with medium or high TMS, so in introducing some sort of medium or even high level of neural noise, they observed a reduction in performance. So there was a classical, let's say, virtual lesion effect. On the other hand, in the low coherence condition, participants show a completely different pattern of results. So in the no TMS condition, here you will see low performance. You will see no effect of medium or high TMS. So if you introduce a lot of noise, this does not really change performance. But if you introduce the low TMS stimulation, so low level of noise, this created a paradoxical facilitation. So introducing just a little amount of noise made participants better in making this discrimination when they were processing low coherence stimuli. So this really means that the optimal level of noise that can be introduced with TMS in the specific region that is playing a role in a specific task can in principle improve performance. And this study by Schwarzkopf was quite nice demonstration of this principle. Well, other important issues to be considered in TMS research is that the effect of TMS can be state dependent. So the same stimulation can produce different effects depending on what the subject is doing. So it can introduce inhibition or facilitation depending on the state, the functional state of neurons in a specific moment. And this can be used. This principle can be used to study to, let's say, improve the focality of the stimulation. I will provide you an example, quite powerful example from the literature, talking about the so-called TMS adaptation approach, which is quite an interesting approach. So here you will see again the V5 area. As I told you, this region is important for motion discrimination. And here there are population of neurons that are directionally selective, suggesting that there are some neurons, for example, the decoding upwards stimuli, or other neuron decoding downward stimuli. So they are different population neuron taking care of different direction of moving objects or just dots, as in this example. For example, here you will see, well, this different, this is of course a schematic representation of what happens in a given bunch of neurons. But let's imagine that in a given volume of neuron, there are both neurons that are selective for downward stimulation here, and other here, these are represented in red. And there are other neurons, so distinct neuro, but within the same area. For example, this could be a voxel from fMRI, so containing actually billions of neurons in this very small volume. And here there are other population of neurons, for example, neurons that are sensitive to upward motions. So, and when you present one stimulus or the other, basically there are different neurons that respond in that specific region. But what happens if you present the same stimulus over and over again? So we know from different neurophysiological study and also fMRI study that when you expose participants with a specific stimulus and you repeat this, there is a decrease in the physiological responsivity of these neurons. So these neurons tend to respond less. For example, these neurons that are colored in red, that were responsive to the downward motion, if you present after several seconds again the downward motion, they tend to respond less. Here, this has been represented in this scheme with a less intense color. And the same holds true for upward motions. So here you will see quite intense green, a vivid green, and here after presentation, these green neurons tend to respond less. So this idea that physiologically neurons tend to adapt can be used to increase the focality of the stimulation. Why is that? Well, because this has been demonstrated several times. It seems that TMS is able to stimulate differentially neurons that are very active and neurons that are less active. So, and this in this particular studies, for example, this has been demonstrated by applying TMS when participants were observing some target stimuli, so showing leftward or rightward motions on one side, the other side, one direction, sorry, or the other direction, and participants had to discriminate these two stimuli. They were very, say, not very, they were brief stimuli with 80 millisecond presented stimuli. And well, they tended to observe this target stimuli after an adaptation to leftward or rightward motions. And typically, when your neurons adapt to a given stimulus, this can result in an impairment in behavioral response. For example, this can be shown here in this graph, where you see that performance to congruent stimuli, so the same stimuli that the participants were exposed to during the adaptation phase, this performance was lower relative to the incongruent condition. For example, if participants were exposed to rightward motions, and then again they were they were presented with the target stimulus showing rightward stimuli, they tend to be less accurate relative to when they were presented with the leftward motion. So an incongruent target stimulus relative to the adaptation phase. So depending on whether the target stimulus is congruent or incongruent to the adaptation phase, they tend to be more accurate or less accurate. So is that clear? So here you will see the performance. So this is the physiological response. So the participants tend to show this performance when it is presented with the adaptation stimuli. This adaptation paradigm is basically the reverse of the priming paradigm, where typically you observe a facilitation. And interestingly, in this study and in several other studies, TMS was delivered in the V5 area, which is involved in these, is where these adapted neurons are, and it was shown that stimulation in this area tended to reverse the relation between congruent and incongruent stimuli. That is, after when participants were stimulated over V5, they tend to show higher accuracy for congruent relative to incongruent stimuli. And this is because TMS tend to stimulate differentially neurons that are active and neurons that are less active. And in particular, in this case, they tend to show it is able to behaviorally facilitate those population neurons that are less active due to adaptation. All right, so it's quite powerful approach because in principle, I mean, these neurons are embedded in the very same region, and depending on the adaptation phase, basically you can target more specifically some a population neuron or the other, and all these neurons are overlapping in the same region. So principle is a sort of micro stimulation, of course, let's say a sort of, but still quite quite interesting approach, because in principle you can manipulate with psychophysics. You can manipulate the activation states of specific population of neurons and test their relevance for behavior by using protocols like this one, this TMS adaptation. There are also other versions like TMS priming, because priming is in principle conceptually related to this phenomenon. But the general idea is that the TMS effects are state dependent, depends on the state of neurons at the moment of stimulation. So one quite relevant element, so this all really when when scholars discovered this, they opened basically a new field of research. Basically, TMS can be used not only by delivering a single pulse, but can be delivered in a repetitive manner. So we can deliver trains of TMS pulses, and depending on the way we deliver these trains, we can induce different effects. So we can use TMS in order to induce plasticity. And in particular, this has been shown several, well, this has been discussed for decades in the TMS literature, because now we have reached this awareness that TMS, repetitive TMS, is able to act on synaptic efficacy, because basically is able to induce long-term depression, long-term potentiation. This was just an hypothesis, a suggestion some years ago, but then TMS has been started to be used also in animal models, and now we have learned a lot of information, a lot of physiological information about the effect of TMS. And so in principle, a TMS train can act on the synaptic efficacy and can, in principle, potentiate, makes this synaptic, so this communication between neurons stronger, so we can induce long-term potentiation, so higher synaptic efficacy, or on the other hand, we can use long-term depression, so we can reduce synaptic efficacy with effect lasting for a few minutes, let's say. And in principle, also this can act on the genetic expression of neuron. That is basically the idea of the expression of proteins if the stimulation is repeated over time. And but the general idea, what is typically investigated, is the effect of repetitive TMS on cortical excitability or, say, specific neurotransmissions. So, but what is typically studied is the effect of TMS on cortical excitability. And now there is a lot of evidence showing that the effect of some TMS protocol, for example, low frequency repetitive TMS, typically induce a reduction in cortical excitability. So for low frequency TMS, repetitive TMS, we means stimulations in which a pulse is delivered each second. So for a few minutes, like 10 minutes, 20 minutes, 30, even 30 minutes, you deliver a pulse at 1 Hz frequency, a pulse each second. And this tends to reduce the excitability of the cortex. This has been studied several times in the motor cortex. And on the other hand, typically we observe an facilitation, increasing in cortical excitability when we deliver trains of high frequency repetitive TMS. So basically, when we use, let's say, 5, 10, 20 Hz stimulations, and we repeat the stimulation over time for a few blocks, we can induce increasing cortical excitability. Similar protocols, well, no, actually quite different protocols, but similar effects have been demonstrated using another protocol. So not the classical standard repetitive TMS, but a protocol called theta burst. It's called theta burst because it is made by high frequency stimulation, which is administered every 200 milliseconds, so around 5 hertz. So that is at theta frequency, let's say. So in principle, in this case, scholars tend to deliver pulses, brief trains of pulses at high frequency, delivered at 5 hertz, at 5 hertz stimulation. As you see in this slide, basically you can have the continuous theta burst or the intermittent theta burst. For continuous theta burst, for example, you apply basically TMS, this continuous stimulation for 40 seconds, let's say. With intermittent theta burst, typically you deliver burst of stimulation for a few seconds, then little break, then another few seconds, then a little break. So as in the example. And this different stimulation, as I told you, tends to induce facilitatory effects or inhibitory effects. For example, when applied to the motor cortex, you can observe that high frequency standard TMS or intermittent theta bursts tend to increase motor evoked potentials over time. And you can observe some effect lasting for a few minutes, up to, let's say, 50, 60 minutes, 1 hour, let's say, depending on the duration of the stimulation protocol. On the other hand, the low frequency repetitive TMS, standard TMS, or the continuous theta burst stimulation tends to decrease motor evoked potential over time. So in white circle, they show like sham stimulation, so placebo stimulation. So you don't see really a modulation over time. And well, this, of course, I have to say, and I have to stress that these effects are those typically reported, but the direction of the effect really depends on the state of the target area at the moment of stimulation, or just immediately before or after the stimulation. For example, in principle, you can induce opposite effects if you, for example, let's say you can deliver continuous theta burst over the motor cortex, and then after the stimulation, you ask participant to make some action to contract muscles. This would create an opposite effect of the stimulation. So instead of having a depression, you will see a strong increase in excitability. So all these effects truly depends on the state of neuron before, during, and after the stimulation, just after the stimulation. So this is quite important to remember, because this can explain sometimes some paradoxical effects because we typically, we now know all these sort of effects, but we, in some condition which we do not control for specific factors, what the subject is doing, then we can observe quite huge variability. And so it's very important to control each phases of the stimulation and maybe to manipulate those phases in order to test the effect of the different manipulations. Okay, I would just like to say a couple of things and then we make a little break. Just another example of repetitive TMS protocols. For example, applied over the somatosensory cortex. This has been shown that if you apply a frequency repetitive TMS for a few blocks, after the stimulation, participant can, show enhancement in the somatosensory processing, as you can see here from this fMRI activation. Basically, the same stimulation creates larger activation in the somatosensory cortex. And importantly, this stimulation is able to reduce the threshold. So that this particular case, this study of participants were solving a 2 points discrimination task, and receiving this stimulation enhancement of the somatosensory cortex made participants better in making this discrimination. So basically showing higher acuity, higher performance discrimination in this psychophysical test. And performance improvement correlated with the activation, with the increase, the enlargement in S1 activation during somatosensory stimulation. On the other hand, reduced burst stimulation or repetitive TMS applied over a specific target region tend to show that, I mean, in principle, you can reduce performance, you reduce cortical excitability, but also functional connectivity. This is probably due to the fact that by depressing a given area, you would make this area interacting less with surrounding regions and regions that are connected to that region. And we have demonstrated, for example, by combining fMRI with repetitive TMS, particularly with theta burst, that continuous theta burst tend to reduce resting state functional connectivity. And there are other regions showing that using intermittent theta burst instead, rather, it induces an increase in functional connectivity. So depending on what you do, basically you could observe increase or decrease in the resting state networks. And this is quite important even when interpreting the effects of stimulation, because as I told you in the first lecture, in the first seminar, the effects of stimulation can be site-specific. If you stimulate different brain regions, you would obtain different effects, but are not site-limited. And this is just another example. When you inhibit one area in a given brain region, actually, you would change the way this area interact with other region, and actually you would affect an entire network. And this is quite relevant, really, when we have to interpret the effects of stimulation of a given area. We were discussing about the effects of repetitive TMS and showing that this effect tends to create spreading of activations. And in principle, when using protocols that are supposed to inhibit a given region, then you would see also some spreading of the inhibition, let's say, and changing the way the different regions interact with each other. So the fact that stimulation of a given area can interact with other region has been one source of one element, one ingredient, I would say, of a new protocol I will briefly introduce to you, which is the cortico-cortical parassociative stimulation. We have recently published a paper together with Boris, and we hopefully would make several other researchers together. Because this protocol is interesting because it's a way of testing Debian plasticity. So as you know, Debian plasticity is a quite fundamental principle of neuroscience, and that basically that states the following. So imagine to have a bunch of neurons that are interconnected on each other, and if a given neurons, let's say the presynaptic neuron, for example, this neuron here down, uh, tends to start to fire uh, repeatedly before a post-synaptic neuron, so a neuron that is receiving some connection from the first neuron. If this experience is repeated over time, the Hebbian principle states that there will be an increase in the efficiency of this synapse, of this connection. For example, I think I have a video here. Well, let's imagine to see this neuron firing just before the other neurons, so the presynaptic firing before the postsynaptic neuron. If you repeat over and over this stimulation, this would create an enhancement in the way that the first stimulus, the first neuron, the presynaptic neuron, would activate the postsynaptic neuron. So a change in synaptic efficiency. And in principle, you can use TMS in order to manipulate the two, of course, not neurons, but interconnected brain region containing billions of neurons being interconnected one each other. For example, again, this is an example from the visual system. If you stimulate V5 just before V1, in principle, you can increase the synaptic efficacy of this V5 to V1 connection, and the other way around also. So, well, the idea is that that was our target, and we stimulated V5 after V5 and then V1 with the idea of increasing the efficacy of these temporo-occipital connections. And the results are quite striking because, well, as you know, V5 is involved in motion perception. If you make this V5 more connected to V1 with this brain stimulation protocol, this would make participants better at discriminating motion. And here show with these red histograms showing a reduction in the threshold, the visual threshold, for a few minutes after the stimulation. And of course, there are several control conditions which we changed the order, the stimulation, the two areas, or stimulated simultaneously, or stimulated with a sham stimulation, placebo stimulation. But only the V5 to V1 stimulation was able to increase the efficacy of this connection and make participants better at perceiving motions. We have recently published, we have now hopefully a Nature Communication paper, which is accepted in principle. We are just revising very minor revision, showing that stimulation of STS, I don't know why it's not okay. Stimulation of STS to V5, STS, the superior temporal sulcus, STS is a brain region involved in perceiving face, particularly face expressions, and enhancing the communication between this face region in the superior temporal sulcus and V1 made participants better at discriminating emotional phases. So, for example, here discriminating between a joyful expression or a fearful expressions. And so this is this principle has been replicated in several other regions and network, even motor network, for example, by stimulating the ventral premotor cortex to the motor cortex, this pathway makes participants better at performing small, accurate actions. So it increased manual dexterity. So this is quite a principle that works in several networks. Quite interesting and novel approach. Well, all these studies have been presented basically show that if you apply a single dose of repetitive TMS or this last protocol CC-pass, but in any case, it's always the same, basically you can induce transient effects. So effects that last for a few minutes, let's say, maybe 10, 20, 30 minutes, even 60 minutes, even 1 hour in some cases, but not more. Researchers, especially researchers that started to apply this repetitive TMS in several fields, including psychiatry, has started to understand that if you apply a repetitive session of TMS, so if you repeat the stimulation, so you do not provide just a single dose, but several doses separated by days, even more dose in a single days, but basically you can induce long-lasting effects, so more enduring effects lasting for days, weeks, even months. But this depends on, of course, on the duration and repetition of the session of repetitive TMS. And so this is quite interesting because it suggests that if you repeat this stimulation over time, these cumulative effects may be also supported by structural changes that are induced over time. And now there is several evidence, and I will start to discuss a little bit about evidence from clinical neurology or psychiatry. So at least I would make some example. And I will start from a study from our lab that was carried out in stroke patients. So we studied basically the effect of stroke on the motor functions, particularly by considering a theoretical model, which is, I would say, a simplified model still, but still can explain several empirical evidence. Of course, at least can explain part of this evidence. So these simplifying models is the idea. It rests on the idea that the two hemispheres are in reciprocal inhibition. So there is a balance between the two hemispheres, the left and the right hemisphere, and the homologous region sends inhibitory projections on the homolog area. For example, the primary motor cortex on the left side send inhibitory projection on the right motor cortex on the right side of the brain, so the right hemisphere. So what is the effect of a stroke in this model? Well, if you have one lesion over, let's say, for example, the left hemisphere, this would create an imbalancement. So this would create an interference with the balance. So, for example, there will be less neurons, less inhibitory neurons on the left part of the brain, and this would create less inhibition on the right hemisphere, because you have less neuron because neuron died, and so they would send less inhibition on the other hemisphere. This in turn would create an enhancement of activity. So at the end, the right hemisphere would start showing higher activity because it's receiving less inhibition. And in turn, right motor cortex neuron would start to inhibit more the left motor cortex. So basically, with this condition, this would the left motor cortex in the affected hemisphere would be in this condition, which have this region has less neurons and receives more inhibition from the other hemisphere, from the healthy hemisphere. So in principle, what we have now learned, what we have learned from the seminar and all the literature, of course, is that in principle you can use a couple of strategies for dealing with this imbalance. For example, you could, in principle, increase activity in the affected hemisphere or reduce activity in the intact hemisphere. And this would be created, for example, by using the protocols that we have discussed a bit. In principle, there are also other protocols, for example, with electrical stimulation, but are not the focus of this talk today. So, in principle, a stroke, there's the interhemispheric interaction. So a stroke occurring in the motor cortex would create an imbalance. Principal TMS can be used to restore at least or try to rebalance interhemispheric interactions. Of course, this could associate with some neuroplastic effect, so an effect on neuroplasticity. This important, the fundamental capacity of our brain to change as a function of the experience that we have. But of course, I have to say that this stimulation is quite gross. I mean, this artificial stimulation, exogenous stimulation that is delivered with transcranial magnetic stimulation, of course, is something very specific. So, while, for example, the neurorehabilitation, so what participants typically do during stroke rehabilitation, that is motor trainings, these are different strategies that act on specific motor networks. So if I do a training, I train some motor function, this would create some network-specific activation. So, I mean, TMS is associated with unspecific motor activation, while motor learning, motor training is associated with specific motor activation. So, but the idea is, well, the question is how these two approaches can be combined, whether, I mean, a very important question is whether TMS can be used to boost the effect of motor learning, the effect of neurorehabilitation. So one, I think, still critical question is how to combine TMS with other techniques that could be classical rehabilitation, psychotherapy, virtual reality experience, or use for some sort of rehabilitation, or drugs, for example. So all these questions are very important, especially for a well, not only for a clinical perspective, but also for a theoretical perspective. But of course, for clinical perspective, they have they can be very useful. So we asked whether TMS could indeed affect neuroplasticity and boost the effect of training in patients with a stroke affecting one hemisphere or the other, so it was unilateral. And so they these patients have a very mild motor deficit, so they were hemiparetic. So they were able to move their affected hand. Of course, they had less force. They were less. They show with less dexterity in the affected hand, but they still could use a little bit. In this graph, you would see please let me say also that participants receive 10 days of stimulation every day. Every day for 10 consecutive days, they receive a daily session of repetitive TMS, particularly inhibition of the healthy motor cortex, of the intact motor cortex using low frequency RTMS. Okay. And then we tested participant just after these 10 days of stimulation. Basically, it was 2 weeks of stimulation almost. Then after 1 week, after 2 weeks, after 1 month, and after 3 months, 3 months after the end of the training, of this training combining both TMS and physical therapy, that is rehabilitation. So importantly, we tested several motor functions. Some motor functions were trained. For example, we train this particular grip, which in hemiparetic patient, it can be very useful, the so-called key grip, for example. So they had to move objects. So this was a goal-oriented rehabilitation. So the participant had to, this stroke patient had to make daily action with small objects to train dexterity and the force of this grip. Okay, and there were 2 groups of real stimulation. In one group, participants received first TMS and then physical therapy, that is the motor exercise, this neurorehabilitation made with a physiotherapist. And another group received physical therapy, and then immediately after, they received repetitive TMS. So there were 2 groups. In total, there were 30 patients, and some received TMS, active TMS, and then physical therapy, or physical therapy and after TMS. And there was also a sham group receiving the same physical therapy, but no TMS at all. Okay, they received sham stimulation. Here you will see that. So this is a tip pinch force. So the force of this pinch. This was not trained in the experiment in this study, this clinical studies. But you will see that even if this function was not trained, TMS groups real TMS groups show an enhancement of the force of this grip that was quite increased in the past and then tended to decline over time, right? But they both groups show an enhancement in this force, in this performance in this task, and they were showing greater improvement relative to the sham group. So the sham group showed some small enhancement at the post, also in the first follow-up, but then tended to lose all the beneficial effect of training over time. After 1 month, it was completely returned to the initial state. Oh, please let me say that these patients, of course, were chronic patients. So they already received a lot of rehabilitation in the previous years. On average, there were at least not at least, on average, were at 3 years from the stroke event, from the ischemic or sometimes hemorrhagic events. So 3 years means a lot. In principle, they are in a very chronic stage. Still, they show some improvement in the sham stimulation, improved a little bit, but the active stimulation improved much more. But please let me stress that this is what happened with those functions that receive no training, right? Let me show what happened with the training with other functions. That is, for example, hand dexterity. But we had also the force of the other grip, the one we trained over time. So do we use different tests? This is just some example. Here you would see that the sham group improves, and the two groups, repetitive TMS and then physical therapy, or the reverse, physical therapy and then repetitive TMS, they show an improvement. In the post and the first follow-up, they showed a similar response that was better than the response and the performance of the sham group. But over time, they tended to show a different performance. And here are the results. Here, as you can see, the sham group, so those receiving the classical neurorehabilitation, tended to show initial improvement, but then they tended to lose the improvement over time. So after 1-3 months, they were basically returned to baseline. And here you would see the two active groups. This group, the one receiving first physical therapy and then repetitive TMS, started to show an improvement at the beginning, but then they tended to lose this improvement. So there was a decline over time, as we observed in the previous graph with the untrained function. Right? So we replicated the same trend in this group, in this group receiving physical therapy and then repetitive TMS. On the other hand, most importantly, we observed that those people who received TMS immediately before physical therapy, they tended to show an improvement, quite conspicuous improvement in hand dexterity, and this improvement lasted for at least 3 months. So there was no decline. I had to say that the physical therapy, repetitive TMS, this orange group tended to show a decline, but still was different from baseline after 3 months. But this group receiving first TMS and then physical therapy was the one showing the strongest improvements that lasted for 3 months. Here you would see a small decline, but at least not significant decline. Of course, I would imagine that after a few months more, if this patient tended not to repeat this treatment, probably they would show a decline, of course. But this suggests that repetitive TMS before physical therapy can induce stable effects after 3 months, at least for trained functions. Using a reverse combination can provide some similar response at the beginning, now just after the stimulation, but then some decline over time. While TMS can show only, when sham TMS, so particularly receiving only the physical therapy, but not a real active stimulation, showing only very transient effects. I repeat, these were patients suffering from chronic stroke, from a stroke, and they were in a chronic stage. So what does it mean? Well, this would suggest that TMS overall can induce some general improvement in motor function across different motor function, but this would likely tend to decline over time. On the other hand, TMS can favor use-dependent plasticity, so can boost the effect of physical therapy, of this positive effect of neurorehabilitation if neural networks involved in engaging in the task are primed just before making physical therapy. So suggesting also that the combination of RTMS and physical therapy can optimize consolidation because they tended to show an enhancement over time that was consolidated even after 3 months. Of course, these long-lasting effects were likely due or were at least partially explained by some virtuous cycles. If the participant realized that their hand was working better, they tended to use more in daily life. This probably would contribute to stabilizing the effect. But still, this is quite promising approach, I would say. Of course, we need replication in larger group of patients, but at least this approach has been actually replicated, and now there is some evidence that combining TMS with physical therapy, especially in the acute phase of stroke, so more acute than the one I show you here, because here in this case we're very in a chronic stage, can be very beneficial for patients. So in the area of neurology and psychiatry, there have been several examples. I show you just a few examples, but I think these are quite promising. For example, in Alzheimer's disease, a colleague of mine, an Italian colleague of mine, Italian group in Rome, has recently shown that repetitive TMS of the precuneus regions. As you know, the precuneus is a say posterior parietal, medial parietal region that is involved in several functions. For example, episodic memory, mentalizing, several cognitive functions associated with projecting ourselves in the future, remembering past events, and so on. This region is altered in Alzheimer patients, so we know this from several studies. And basically, in this study, a group of I think were 50% with Alzheimer's disease were assigned to different groups, one receiving TMS, active TMS of the precuneus, and another group receiving TAM stimulation of the precuneus. And these receive a daily session for 2 weeks. So daily session of TMS, either repetitive, actual, real repetitive TMS or stimulation in the intensive phase, and then they tended to receive further stimulation at a lower rate. So instead of having daily session, they had weekly sessions, right? So they receive in total this. They receive with less frequent stimulation over 22 weeks, right? So quite long, prolonged stimulation. And this is quite important because especially with neurodegenerative stimulation, but not only neurodegeneration in general, if you want to observe a strong effect over time, then you would need to consolidate. You would need to consolidate by repeating over and over time. We have learned this especially from studies on depression. And I will talk about this in a few minutes. So here you would see the effects, which are quite promising. For example, you would see this clinical dementia rating showing, for example, that those who receive active stimulation, they also show a maintenance of their performance over the 24 weeks over time. So they show with a quite stable clinical performance, while the sham group over this period of 24 weeks, they tended, so basically half year almost, they tended to show a decline in this task when this clinical assessment. So we observe they observe similar effects with the ADAS, the cognitive scale of the ADAS, the Alzheimer's disease assessment scale, or the mini mental examination test, or these activities of daily life, daily living scale of this clinical assessment test. And it was striking because all these Alzheimer disease patients tended to show decreasing performance over time, while those receiving this stimulation remain stable over time. Here you would see physiological effect of the stimulation. They use TMS and electroencephalography and show the effect of the stimulation of the precuneus over the rest of the brain. And basically, this showed that the stimulation over time showed stable effects in the active stimulation group. So after 24 weeks, they show basically the same activation as in the initial state before any repetitive training. And while the other group tended to show reduced activation state over time. So the effect of the stimulation of the precuneus was lower 24 weeks after the stimulation. This is another, so we do have now several clinical trials like this one and the previous I presented, where in still small group of patients, but not so small, sense that some dozen of patients, typically showing with double-blind procedures, showing better performance or at least preserved performance over time following this treatment. So now there is really an explosion of applications in different conditions, not only Alzheimer, but also Parkinson. Actually, Parkinson is one of those clinical conditions that have been studied more, and other pain and other conditions I will to discuss in a few slides. But before discussing further about the clinical effects of TMS, I would also like to mention, of course, some safety issues, of course, because Boris, you asked me to say something about it, and so I'm happy to say a few things. Because the first thing, of course, when using TMS is not to harm, of course, no, to induce benefic effects, not negative effects. And this, of course, is this is, well, some joke, of course. But I have to say that in general, TMS is safe, is a safe technique and well tolerated. However, it should be considered that the high frequency repetitive TMS is associated with some low risk of inducing seizures, so epileptic activity. And so this is quite important. This is a serious issue to be considered. And so in a safety guide, the international safety guideline published a few years ago, there were reported a few examples of basically patients that showed seizure after high frequency TMS, not only high frequency TMS, but there is a consensus in identifying which are the conditions that can that create a risk for inducing these negative events. And typically, these are associated with specific factor that has been reviewed in several international guidelines, recommendation, etc., that include, for example, the assumption of specific drugs and basically all patients showing some epileptic activity, typically they were taking some specific drugs for through cycles, for example, or they had some stroke and they had some brain condition that that increased the risk of epileptic seizures. So I would, for those who are interested in the safety issue, I would recommend to review some of the expert guidelines. Those by Simone Rossi. The first was in 2009. Another couple of years ago, in 2011, was published a questionnaire for ruling out condition. This is quite useful for those in Talca who would like to start using TMS. It's would important to screen participants with a ruling out specific condition that are associated with seizure or eventually with, in general, with issue associated with TMS stimulation, like, I don't know, for example, having some pacemaker or metal implant over the head and these sort of things. So I don't know what time is it. Oh, it's all time is almost over. Well, I would just mention very briefly that there is and now there is an explosion of interest in clinical application of TMS and other brain stimulation techniques. In 2008, the Food Drug Administration in the United States approved a treatment for patients suffering from depression, specifically high frequency repetitive TMS. And since now there are several other FDA approved treatments, for example, for pain, smoke smoke use when they want to smoke cessation when they want to quit smoking, actually, obsessive compulsive disorders, some anxious, depression, comorbidity, pain, chronic pain, and other conditions that are really so in the psychological and say in the clinical psychiatry, in the psychopathology field, there are now several applications. So not only in the in neurology, but also in psychiatry and other conditions, for example, physiatry and so on. So now I would recommend for those of you who are interested in the therapeutic use of repetitive TMS, there are also in this case several meta-analyses in the literature Now there is another group by the, very, very important scholars in the field. I would recommend to have a reading of evidence-based guidelines for therapeutic use of TMS. We'll finally consider all the advantages and disadvantages, the positive, the pros and cons of TMS. We can consider the first important issue for those who are interested in cognitive neuroscience is the possibility to test and investigate causal links between brain structure and function. So establishing causal link between a brain region and a cognitive process, perceptual, cognitive, motor process. So TMS can have an excellent temporal resolution. For example, a single pulse stimulation can induce effects that last for a fraction of millisecond, maybe creating some spreading lasting for a few milliseconds, but really you can reach very high temporal resolution in testing the functionality of a given brain area over time. When delivered in a repetitive manner, TMS can induce neuromodulatory effects that last for a few minutes after the end of stimulation. And as we have discussed by discussing about stroke and also Alzheimer disease examples, we can even induce longer lasting effects when using repetitive sessions of stimulation, repetitive application. So we can have a very good spatial resolution, but this depends, as we discussed in the first seminar, on the size of the TMS coils or on the intensity. If we use higher intensity, we recruit larger portion of neurons. If we use smaller intensity, we recruit less neurons. TMS can provide an excellent combination with other techniques like electromyography, if we are interested in recording motor evoked potentials, or in electroencephalography, if we're interested to use TMS evoked potential. These are quite important informative measurement. Then last two positive aspects, the low cost. So, I mean, comparing to MRI or PET or magnetoencephalography, of course, TMS is quite cheap, cheap techniques, maybe less, not that cheap as electrical stimulation, but more, I would say, more promising for both research and clinical applications. And of course, it has some promising clinical application. Now, at least in Italy and other countries, now there are opening several clinical centers that offer TMS for patients. So now there are several examples, my countries and several other countries of clinical center open to patient that pay to receive TMS, combined with maybe other treatment as well. And negative aspects, of course, for those who want to study the effects of stimulation of specific brain region, I mean, we have some limitation because we cannot reach deep regions, although now people are using different coils trying to reach more deep region, but still, even with these deep coil solutions, actually what is stimulated more is always the superficial part of the brain. So, I mean, sometimes people talk about deep TMS stimulation, but actually I would call large TMS stimulation because of course it's can reach deeper areas in the brain, but also this is associated with the larger recruitment or more region of the superficial part. So but still now, I mean, there are some anatomical limitations. Well, we can use a correlation approach. We study TMS, evocative potential, etc., but we are limited still to superficial areas. We lose spatial resolution using high TMS intensities. And well, it requires appropriate sensitive task and when this well-designed paradigm. Forget to observe dramatic effect with a single dose of TMS. With a single dose of TMS, we can, for example, just let me provide this example from, just to create an idea. We know, for example, that a lesion over right parietal cortex can induce neglect. Neglect is a hemispatial neglect, which is a neuropsychological condition in which participants do not attend the left side of the space. And for example, maybe some patients do not focus attention on their left hand or the left part of the face. For example, when they shave, they forget to shave on the left part of the face. Well, forget to induce some effects similar to that, to these real symptoms that they observe by lesion of the right parietal cortex. Forget to observe something similar when you disturb the right parietal cortex with TMS. Of course, you would maybe using a very sensitive task, for example, trying to find, for example, the middle part of a line, of a horizontal line, which is a quite sensitive task. If you neglect the left part, you tend to put to bisect this line over the right part. And of course, maybe this task is quite sensitive. You will see an effect of right parietal cortex stimulation in this condition, but you will never see someone who forget to shave on the left part of the face. So it has some limitation, of course, and of course we should consider, especially in the research setting, that online TMS can be distracting, and sometimes, depending on the region of the scalp, can be unpleasant. For example, when you stimulate ventral region, this can create some muscle activation, which can be considered unpleasant. Not really unpleasant, but some participants. Some participants prefer to receive less intense stimulation, let's say. So let me thank my group, my students, and collaborations all over the world. And thank you very much to you for your attention.